All right, hello everyone and welcome to this Hybrid Ag webinar. My name's Sarah Tinsley and I'm a member of the Hybrid Ag Research and Support team. I'm excited to introduce this cell expansion webinar, which is the third part in a 6-part series of events focused upon the stages of plant nutrient importance scheduled for this year. Through orchard walks, field trials and webinars like this one, we'll take you on a graphic journey through this season, looking deep into the technical aspects and scientific facts about soil health and plant nutrition. We know that you're all busy, but we think there's significant value in taking time to take a step back from our businesses in order to better understand exactly what is happening in your soil and in your plants in order to have healthy soil, healthy plants, nutrient-dense food, and of course, bigger profits. Hybrid Ag are on a quest to transform food for health. That's our purpose. And during this journey over the past 20 years to discover more about what makes food healthy and how we can grow better nutrient-dense food, we've curated over 150 years worth of soil and plant nutrition research and data. Today, we have what is probably one of the world's most comprehensive databases in this field, and we'd love to share that with you. Many of the subjects we'll be covering through these events will be quite technical, and we don't make apologies for that. We will try to present these subjects in a way that's easy to understand, but if anyone would like to discuss any aspect of these events with us or their contents, then please get in contact with us where we'd be delighted to answer any questions that you might have. So I'll introduce you to the panelists that we have today. So first up, we've got Hamilton Rowe. So Hamilton Rowe is one of our soil and plant nutrition scientists specialising in pome fruit, stone fruit, cherries, herbs, nuts, and citrus. He has added significant value to growers over the past 6 years, creating custom blended soil and plant nutrition programs for Hybrid Ag. Hamilton is also a founding member of the Transforming Food for Health certifications. And now many of you will know Andrew Smith. He's a fruit grower and soil and plant nutrition specialist, and he works with cherries, stone fruit, pome fruit, table grapes, and citrus. He's enjoyed over 35 years running his family's stone fruit business. The Andrews family has been growing stone fruit for over 150 years, and he has been working with Hybrid Ag for the past 8 years as one of our leading tree fruit specialists. And me, my name is Sarah Tinsley. I'm a Hybrid Ag soil scientist working at the Research and Support Centre in Wangaratta. I coordinate educational material like this webinar. I'm currently completing an honours year in soil science, and I have a passion for making scientific information usable to everybody. So if you have questions throughout the course of this webinar about any of the content, please feel free to leave them in the Q&A box at the bottom of the screen, and we'll be able to address them at the end. So now that we're all introduced, Hamilton, would you like to kick us off?
Thanks, Sarah. So just to put today's topic into context, I just wanted to explain exactly where we'll be focusing on in the season today. So you might notice that for those that joined the first webinar, the webinar series has expanded a little because we decided there was some more room for specific webinars to cover stages of nutrient influence. We've now divided the webinar series into the 6 listed here. So this is webinar 3, focusing on cell division. So different nutrients are in focus in each stage, and therefore it is important to understand the differences between the stages so that the nutritional needs of the plant can be met consistently. High quality yields come when the plant does not experience any nutritional stress or deficiency across any of these stages. If you're interested in learning more about the other stages and their optimal nutrient management, feel free to get in touch with us or follow our social media pages for more information on our upcoming webinars. Just move over to the fruit fill phase. So that's it's also the cell expansion phase is what we refer to it as. This is where cells that are created during the cell division phase become larger. So it's purely about enlarging the cells that are created during cell division. So cell expansion is a vegetative growth phase, and it's driven by a slight dominance of the plant hormone auxin, which is a vegetative hormone. Cytokinin, which is the reproductive growth hormone in the plant, is still present and required in this time, but the balance just becomes slightly tilted in favour of auxin dominance. Nutritional management at this stage influences the quality of plant cell growth and can improve fruit or crops in terms of several key quality parameters such as fruit density, size and weight, firmness, skin finish, and shelf life. So in this webinar, we'll be discussing 4 key topics that are specifically relevant to cell expansion. So the first one is how a cell is expanded, then the below ground support for cell expansion coming from the roots, the above ground support for cell expansion coming from the leaves, and then specific nutrient requirements which are applicable to the cell division phase. So let's get started with how cells are expanded, and I'll pass over to Andrew.
Thanks, Hamilton, and welcome everybody to today. Okay, so we've got a picture of an actual fruit cell here, and it's cross section, as you can see all the little bits and pieces which belong to the cell. So you can see the vacuole and the cytoplasm, which is the yellow part which surrounds the vacuole. So fruit cells are expanded as water and phytoassimilants flow into the actual fruit, which is sugar complexes and amino acids. So water goes into the cytoplasm and the vacuole of the cell, rapidly expanding the actual vacuole. So the vacuole is a storage sac of the cell for enzymes, fatty acids, sugars and amino acids. And it also works with the waste products which are produced within the cell. The cytoplasm is actual fluid that surrounds the vacuole. So as the cell wall is required to give way to the pressure produced by cell sugar, this is the osmotic pressure created within the cell, which creates the water movement throughout the plant. So calcium is important to maintain energy and nutrient transfer through the cell wall to facilitate growth and actual cell expansion. The plant will favour the growth of new shoots over the growth of the fruit at this particular time. It's important to note that the nutrient applied at this time goes towards maintaining cell growth within the fruit rather than the actual new shoots that are growing. So this is where nutrient balance is very important within the plant and must be maintained. So we'll move on to section 2 now, which is the below ground support. So we want to talk about the 2 different types of sap flows which move within the plant. So we've got the xylem and the phloem sap flow, which you can see there in the slide. So one's an upwards movement and one is a both directional movement. So the cell expansion is driven by the movement of water and phytoassimilants throughout the plant, and it's also important to understand how these are being transported. So a phytoassimulant is a carbon compound that is created during the photosynthesis cycle, for example, sugars and amino acids. Water movement into the cell has been researched extensively, and whilst a large percentage of the water movement is moved into the fruit through the phloem, the xylem plays a vital part in this process, providing water throughout the fruit as well. So the xylem movement occurs in one direction and moves throughout the non-living cells carrying the water and minerals. So this is only an upwards flow of the water and minerals into the plant. Whereas the phloem, on the other hand, largely carries the complex solutions back down the plant to the fruit and the root sinks, which contains the organic molecules, amino acids, and the other second metabolites as well. So the water flow in this particular pathway flows both ways. So the cells which make up the actual phloem are living and also required support from the plant. So this is this 2-way movement of the sugars and minerals within the plant. So different kinds of roots will contribute to these 2 types of movements. So if we go to the next slide, we see our primary and secondary roots. So when we look at our picture of our roots, we can see what's commonly known as a tap root, which we classify as the primary root, which is where carbohydrates are stored. And then our secondary roots or lateral roots, which are there for movement of sugars and water as well. So when we look at the importance of root growth and cell expansion, we can see there in our next slide. So importance of the root growth of the cell expansion. So primary roots and their functions, and secondary roots and their functions, and what they do in the early part or mid to late part of the actual growing season. So the root growth is particularly important for cell expansion. So primary and secondary roots are both required at this time and serve similar purposes, but at different times. So in reality, the process is much more complex, but we have simplified it for today's purpose for the webinar to just make it a little bit more easier to understand. So the plants are supported by carbohydrates supplied from the primary roots early in the growing season, which has been stored from last year's post-harvest program and the plant senescence cycle. So the balanced nutrients are critical to maintain the hormone balance within the root zone, and there is a slight oxygen dominance through the cell expansion phase, which also encourages the second root growth and the photosynthesis process. So when we talk about hormones in the root zone, the auxins actually stimulate the hydrogen within the root zone, creating the organic acids, which are critical to provide the nutrient uptake into the plant and stimulate the secondary root production. So excessive N can lead to significant reduction of the cellulose and lignin within the root tissue that can inhibit the secondary root growth. This can also increase disease and insect susceptibility within the root zone, which affects the efficiencies of the whole plant cycle and impacts the actual cell expansion phase. So the keys to remember is that the health of the root system determines the outcome of the actual cell expansion. So maintaining the healthy root growth is about the correct hormone balance, which largely will achieve the correct nutrient balance within the plant. So we'll now move on to section 3 of the ground support, and I'll hand over to Sarah and she'll talk about photosynthesis.
All right, thanks for that, Andrew. So I can see that we've got some great questions coming in. So just a reminder that everyone, you can use the Q&A box at the bottom of the screen to submit any questions that you might have throughout the webinar. So back to photosynthesis. After the carbohydrate stores from the primary roots are used up in early cell expansion, the energy is sourced from photosynthesis. So photosynthesis begins as water is brought up through the roots of the plant and is combined with carbon dioxide and light taken in through the leaves. Using the energy of the sunlight, water and carbon dioxide are transformed into sugars that are used by the plant and an oxygen gas that's released back into the air. Using this example, the leaves can be imagined as solar panels, providing the plant with energy to run. However, throughout the growing season, the kinds of leaves providing these sugars for fruit growth change. So I'll pass back to Hamilton and he'll be able to explain these different kinds of leaves to you.
Yeah, thanks, Sarah. So understanding how leaves support the cell expansion phase is extremely important, especially in tree crops. So this graph we see here shows a relative contribution of the different leaf types after bloom, specifically in apple trees. So apple flowers opening in the spring are supported by tree carbohydrate reserves from primary roots, and that happens up until bloom, and then post bloom the fruit growth is supported through the current photosynthesis of the leaves. So this same process it happens in all tree fruit crops as they come out of dormancy. So firstly, cell division is carried out by the spur, the cell division support is carried out by the spur leaves in the critical weeks during that phase, and then they also support the first stage of cell expansion, as you can see in this graph. So the spur leaves are largely fueled by the carbohydrate reserves that are stored in the plant, and then they're also supported by early secondary root growth. However, what happens is you end up with carbohydrate stores being quite low after cell division. So as we come into cell expansion, this is where applied nutrients can make up the gap, maintaining high energy levels in the plant to maximise the photoassimilates, the secondary root growth, and the phloem uptake within the plant during this period. So in apples specifically, as shown in this graph, the spur leaf productivity declines, and then the red dotted line is where the bore leaves take over, which they become the dominant source of energy to grow the fruit until about 45 days post bloom. Bore leaves are those that sit on the short lateral shoots on the spur. These leaves bridge the gap between the spur and the shoot leaves before quickly declining their actual use as part of the cell expansion phase, as you can see by that red line. This role is played by the first leaves created post-flowering in other fruiting plants, which are at the base of the new shoots, but not part of the actual shoot growth itself, and are not spur leaves. So in this case of apples, finally, after about 45 days, extension shoots become the main energy source for cell expansion. So extension shoots do not support fruit initially during their initial growth stages. So that's why their peak of support for the fruit doesn't actually come until probably about 60 days, as you can see in this graph, after bloom. So these periods are obviously slightly different depending on the fruit crop and depending on the length of the total fruit growing cycle, but the same process happens specifically between the spur leaves, the first leaves or borse leaves, and then the shoot growth. In other crops, such as vines and berry crops, normally there's plenty of leaf prior to cell expansion, so the roles of specific leaf types are different. However, the nutritive processes, which we'll go over, that are required for this cell division or cell expansion period are exactly the same. So we'll move on to looking at growth rates over time. And this is just to emphasize the importance of early fruit growth and how much of an impact that has on the overall size of your fruit in your ability to expand the cells. So early fruit growth typically has a pattern that looks similar to the graph that we've got here on the left, which is in tomatoes. So it begins quite slowly in the first few days after the growth. It really starts to steeply increase and grow very quickly. It reaches its maximum growth rate in the first 35% of the growth days after cell division in tomato plants. And that obviously there's research to show that this applies across the other different crops, such as apple and citrus. So applying this principle, you see it gives apples approximately 32 and 36 day max growth periods, which is the period that's highlighted in orange here in this tomato graph. And these periods are about, in apple and citrus, probably around 80 to 100 days post flowering. And then obviously after this, the growth rates drop quite significantly. So it just shows that early period, how critical that is for maximizing your cell expansion potential. And then as you would well know, the nutrient applications then need to actually be applied a good 25 to 30 days prior to where this growth rate starts to increase, to ensure that the plant reserves of each nutrient are abundant and available for when the plant starts to use them. So I'll move to the last stage of the webinar, which is obviously going over the specific nutrient requirements during this phase. So fruit fillers, all stages do have a specific set of nutrients which are required. First and most importantly, nitrogen is required to maintain an oxygen dominance, a vegetative dominance, and increase secondary root growth and shoot growth. But it's just, you've got to be mindful that it must be kept in balance with the other nutrients. Potassium is required to maintain effective transportation of carbohydrates and sugars around the plant and sugars to the fruit. So it's used in the osmotic process of water transfer, as Andrew touched on earlier, and the turgor generation. So which has a direct result on the ability of the plant to expand the cells. So then iron is important at this time because it's used for the early photosynthetic cycle of the first leaves, and it's also important to put together the chlorophyll molecule as part of the photosynthesis process. Manganese is crucial during this period as it's used for water hydrolysis in the plant, and water hydrolysis occurs in the beginning stages of photosynthesis, which means that the photosynthetic processes can be reduced if we have low manganese environments. So it's extremely important to have very available stores of manganese in the plant during this period. Zinc is also very important as it is key for the production of oxygen molecules, and it's also a cofactor for ATP, which is the energy carrier within the plant. So ATP is necessary for converting sunlight, carbon dioxide and water into plant usable glucose, which is essentially the beginnings of, or the basic process of photosynthesis. And then boron, fundamental in this stage for moving and directing sugars around the plant to the different sugar sinks, which is key in this stage of cell expansion to move the sugars towards the fruit. A note to make here is those 3 points. So manganese, zinc and boron all have a relationship with the production of oxygen. So that's also an important thing to take note of during this stage when we're trying to maintain slight oxygen dominance in the plant. Copper is also required during this time to maintain strong stems, skin elasticity. So in soft fruits especially, we've observed a strong reduction in the amount of cracking or splitting that we see in fruit through adequate supply of copper in the cell structure. So copper is important in the production of lignin, which is the organic compounds in the cell walls to form strong, rigid cell walls with rot resistance. So another part of copper is it's also has a natural antimicrobial properties. So it's also extremely useful for pathogen resistance. The other thing which is not touched on this slide, which we went over in the cell division webinar in quite a lot of detail, is that calcium supply early is extremely important for your cell integrity, cell wall integrity and structure, and obviously no different through this stage. As the plant starts to kick into photosynthesis, we should be having abundant supplies of calcium coming up through the root system, and so it's just important to maintain that supply of calcium through this stage as well to maximize our cell expansion. Obviously, as always, when considering nutrient applications through any stage of plant growth, but specifically in this stage, it's mindful to never focus on one nutrient over another. They all play a very important part in the outcomes of cell expansion. So I'll pass back to Andrew to go over some summary slides.
Okay, thanks Hamilton. Some really good information of the above ground support and obviously the nutrient requirements there as well. So when we look at the plant health, and we've been talking about the balances, so throughout this presentation we certainly repeatedly referred to plant balance. So the balance is needed between all these examples which are listed on this particular slide. So you can see from photosynthesis to the nutrients present within the plant, the actual plant hormones, based around cytokinin and auxin, and then our primary and secondary root functions and also the growth within those. So these balances are very delicate, and small shifts during this time can have significant impacts on our final fruit crop outcome. So as growers, it's important during all these stages to consider the nutrients that are already present in your soil, the climatic conditions that your plants are exposed to, and the actual nutrient requirements of your crop to maintain these balances throughout the growing phase. So, your water, your nutrient applications must be targeted, and these timings are very critical. So we need to remember that every nutrient will also impact other nutrients. It's extremely important to have the abundance of the balanced nutrients available through this cell expansion phase to maximise the genetic potential of the plant and also to maximise actual plant health. So if we get these balances right, everything else will sort of follow and fall into place. So when we look at the last slide that we've got here based around, okay, so what are our grower take-homes? So the maximum growth rate really does occur in the first half of the cell expansion phase. So it's very important to maximise this stage. The other thing is the spur leaves actually maintain the initial fruit growth and use that stored carbohydrates to drive the early cell expansion and fruit growth. So this is where your primary root really comes into play, working on its energy which has been stored, or the carbohydrates which the plant will use in the start-up in the early spring cycle. So nutrients within the plant should be balanced to maintain the primary and secondary root growth to help maximise our cell expansion. So all balances should really be maintained throughout the growing season to ensure the best possible crop outcomes. So I'll now hand back to Sarah, and she can just go over the final slides.
All right, great. Thanks for that, Andrew and Hamilton. And thank you all again for joining us in our webinar today. In our next webinar, we're going to be following on from today's discussion, covering the stage of bud initiation. This webinar will happen sometime in October, so we'll let you know as soon as we have an exact date for that. If you have any feedback from this webinar to help us improve, we'd love to hear from you as well. So just on the next slide, we have some contact details. So give us an email on support@hybridag.com.au or give us a ring on 5722 7555. So we've had a few questions during this webinar, which we'll cover in a document that will be emailed out to everybody. But just for now, Andrew and Hamilton, would you like to select a couple of questions that you find most interesting out of the list?
Yeah, there's actually some really interesting questions that have come through here. So there's one here. How would you recommend trace elements be applied in season, e.g. foliar fertigation or granular? And then also, do you prefer chelates or generic chem such as sulfates or oxides? So really great question. Obviously, with trace elements, they're the most efficient way that we've found of using these, especially once we're into the season and we've got leaves on the trees and the trees into its full photosynthesis cycle. Using foliar sprays of trace elements is extremely efficient and highly effective. And 100% this needs to be done as a chelated trace mineral pack. Sulfates don't have the same efficiency of actually getting into the plant sap. And oxides obviously end up building up, you build up oxides in the leaf structure, and then it doesn't actually transfer efficiently into the sap flow. So what we've found is the most efficient and effective way is through chelated trace minerals, and the best way to do this or to monitor that is through leaf testing, obviously, rather than just applying a nutrient without actually understanding whether it's needed or when it's needed.
All right, great. Thanks for that, Hamilton. Andrew, would you like to pick a question as well out of the list?
Yeah, okay, sure. Certainly, there's quite a few here. So, do trees grow new secondary roots every year? Well, short answer, yes. So you've obviously got a root mass which is always situated in the soil from year to year, but as your tree starts up, yes, it does push new secondary roots. It's part of the process. It will keep adding new roots to its root system annually each year. So yes to that answer.
Great, thank you for that. And we might just do one more to keep it nice and close to 11:30. So Hamilton, would you like to choose another question?
Yes, there is one here. It says, why do you say to apply nutrients 25 to 30 days before cell expansion? Foliar sprays effective within 24 to 48 hours. So, great question. And the reference that I made there was specifically around when the plant is coming into that cell expansion maximum growth phase, and what you're trying to achieve by applying nutrients early is that they actually get up into the plant and are stored in abundance, so they're available to the plant to essentially naturally pick and choose what it needs at specific times to maximize the outcomes of the cell expansion phase. So that's the thought process behind the applying nutrients earlier and having them available for plant use once you're into that phase.
Great. All right. Thanks for that, Hamilton. So if you haven't asked a question today, but you would like more information, then please don't hesitate to drop us an email or give us a call. All of our questions and answers will be available through our certified partners as well, who are David Morello from Morello Fert, Noel Jones at Sahara and Elders, Craig Swansbury at Fruit Doctors, and Graham McNeil at GB Crop. Finally, thank you to all of the technical experts from around the industry who have helped bring you these events, and a special thanks to Judah Rowe as well for his work in compiling some of the content for this webinar. And I'll see you next time, and don't forget, happy soils make healthy souls. Goodbye.